The next speaker is Dr. Irina Kufareva. She is currently a project scientist with the UCSD Skaggs School of Pharmacy, and she's been there since 2009. She completed her PhD in computer science, actually, in 1999 at Tomsk State University in Russia. Did some postdoctoral work in computational structural biology at TSRI in 2004. And her current interests include computational protein ligand complex structure prediction, particularly for G protein coupled receptors, kind of generally, but specifically and relevant here today, also chemokine receptors. And so today, her talk is entitled Structural Based Chemokine Binding and Specificity Toward the Two HIV Co-Receptors, CCR5 and CXCR4.
So I want to start by thanking the organizers for inviting me to give this talk. It's a great opportunity and a pleasure to be here. And just attending these talks, I like getting the perspective of tackling the challenge of HIV from different levels, starting from epidemiology, and here we are doing trying to understand the atomic level of interactions, molecular interactions involved in HIV transmission and infection. And I want to especially thank Victoria for introducing the concept of molecular docking and the importance of introducing flexibility in this molecular docking. And my presentation will be about the structural basis of interaction of chemokines with two HIV co-receptors. So to start with that, cell migration in humans is critically dependent on the system of chemokines and chemokine receptors. Chemokines are small, soluble secreted proteins, and their receptors are transmembrane proteins with 7 membrane-spanning helices that belong to a larger family of G protein coupled receptors. HIV infection, as it happens, depends critically on this chemokine system, because as HIV virus infects macrophages at the initial stages of infection and then T cells later in the course of development of the disease, the virus does so by first binding the surface glycoprotein GP120 to CD4 antigen on the cell surface that is shown here in green. And let me get my mouse. Yes, CD4, that induces a conformational change in the GP120 glycoprotein and further allows its interaction with one of the co-receptors, the so-called co-receptors, and that would be either chemokine receptor CCR5 or chemokine receptor CXCR4, depending on the viral strain. So that in turn enables the virus to enter the cell. Without CCR5 or without CXCR4, this infection is not going to happen. And consistent with that fact, binding of molecules to CCR5, for example, is one efficient strategy of preventing HIV entry into host cells. And for example, a small molecule under the name of Maraviroc gained approval, FDA approval in 2007 as an inhibitor of HIV entry and as a therapeutic approach for treatment of HIV infected individuals. I'm showing here a structure, actually, a structure of the receptor with the bound Maraviroc molecule in its binding site. So this is the one that prevents interaction of GP120 with the co-receptor. However, HIV is tricky, and HIV develops resistance. And in particular, it can develop resistance to Maraviroc therapy by learning to use different species as co-receptors for host cell entry. For example, it may learn to bind Maraviroc-bound CCR5 species, or it may learn to bind from the entire population and conformational variability of the receptors on the cell surface. It may learn to bind Maraviroc-incompatible species, or if nothing else helps, then it can learn to bind CXCR4. Now, chemokines that, like I said, they're endogenous agonists of chemokine receptors, and they are small secreted proteins. As it turns out, they also hold promise as to inhibiting HIV entry. They do block it, and they actually are less prone to the development of resistance than small molecules are. However, there is a level of trickiness in that as well, because their anti-HIV potency, as it happens, does not correlate with their affinity to the receptors. And instead of that, it frequently correlates with the kinetics of dissociation of these chemokines from their receptors. So slower dissociating variants would be more efficient. And it also depends on their pharmacology. And as this diagram shows here, and that's adapted from a publication by Oliver Hartley, as this diagram shows, the efficiency in inhibiting HIV actually correlates with the ability of the chemokine to internalize the receptor, bring it inside the cell, and keep it there. As such, wild-type chemokines are usually not effective inhibitors of HIV entry, but engineered variants are. And Oliver Hartley's lab in the University of Geneva has been working for a long time on designing such variants. And one of the variants of chemokine CCL5 that they designed is now in clinic as a low-cost vaginal microbicide that actually works by preventing GP120 binding to CCR5. And by the way, Oliver Hartley was presenting for CFAR. Many of you probably have attended his talk maybe a couple of months ago. That's a strange place for acknowledgments, but I want to bring up the fact that this work that I'm presenting is a collaborative effort of a computational lab by Ruben Abagan, and I am a part of that lab, and an experimental and structural biology lab by Tracy Handel, both in the School of Pharmacy. We started that close collaboration some 5 years ago under the NIH funding on structure, function, and activation mechanism of chemokine receptors that now transitioned in understanding of chemokine receptor interactions with chemokine and HIV GP120. Now, in the course of our work, we're asked these questions. How exactly do chemokines bind the HIV co-receptors? And of course, we would like to answer this question by X-ray crystallography. But it turns out that X-ray crystallography is not only challenging, it's especially challenging for proteins that are embedded in the cell membrane, such as chemokine receptors. And that's why that work is being complemented by molecular modeling. Now, when that question is answered, and I will not cover questions 2 and 3 in my presentation, but in long-term perspective, what we want to understand is the structural basis of flow kinetics and diverse beneficial pharmacology of chemokine variants that makes them good HIV entry inhibitors. And also, we want to learn to rationally design small molecules with similar mechanisms of action. In terms of understanding receptor chemokine recognition, as of last year, it was based on the so-called two-site hypothesis, which said that recognition between the chemokine and the receptor involves 2 distinct epitopes. One is binding of the sulfated flexible N-terminus of the chemokine, of the receptor to the globular core of the chemokine, and it is called chemokine recognition site 1. And the other one would be flexible N-terminus of the chemokine binding in the transmembrane domain of the receptor, and that is called chemokine recognition site 2. The blue interaction is known to contribute to binding affinity, whereas the green one is important for both affinity and signal capacity of these chemokine variants. We would like I said, we would like to understand the structural basis of the interactions by crystallography, but with G protein coupled receptors in general and chemokine receptors in particular, it's hard because they typically give you very low protein yield. They're unstable outside their native membrane environment. They're conformationally heterogeneous, and they literally have no hydrophilic surface for formation of crystals. We use a pipeline for screening of pre-crystallization properties of these receptors, and most wild-type receptors would fail at different stages of that screening. They aggregate, they just don't behave well enough to form crystals. And that is why protein engineering is critical. There is a series of approaches that can be used to modify these receptors and make them better behaved and crystallizable. Now, that said, protein-protein complexes add another level of difficulty to the problem because these tend to fall apart even when the individual components are stable and well behaved. And when we think about interaction of receptors with chemokines, that's a protein-protein complex involving a GPCR. Despite the difficulties, earlier this year we published the first ever structure of a receptor with a chemokine, and that is the one on the left. It is a chemokine receptor CXCR4, one of the co-receptors with chemokine called VMIP2. It's a virally encoded chemokine from herpes virus that does not have anything to do with HIV. Nevertheless, it gave us an idea of how chemokines interact with the receptors. And a few weeks after that, a group from Stanford, Chris Garcia, solved the complementary case where human chemokine was solved in the complex with a viral receptor. So on top of that, as a molecular model, a computational molecular modeler, I am working developing hybrid modeling and docking protocols that involve conformational stochastic optimization of a system in internal coordinates, and it is based on hybrid representations of the components that allow to account for some level of flexibility. And as Victoria just told us, flexibility is very important in forming these protein-protein complexes. In addition to that, these protocols allow integration of experimental restraints, for example, cysteine trapping. Cysteine trapping is an approach where we introduce single-point mutations to cysteines at strategically selected positions in the chemokine and in the receptor, and we co-express them in cells in a hope that if these cysteines are in proximity in the native geometry of the complex, they will lock down and form us an irreversible complex. Using these approaches, one of the things that we've done, we built a model of CCR5 in complex with 5p9 CCL5. CCL5 is an endogenous chemokine agonist of CCR5. 5p9 CCL5 is its engineered variant from Hartley lab, and it is different from the wild-type protein by its N-terminal sequence. It is an antagonist. It is a slow off-rate antagonist with potent anti-HIV activity, and it is a close relative of that variant that I mentioned at the beginning that is now in clinic. In addition to its N-terminus, it was discovered that a certain residue pretty far in sequence from the N-terminus was important for anti-HIV activity of this and other chemokines, but not for binding. Now, by looking at that model that I'm presenting here, and I'm presenting it in 3 orientations. So that's kind of front and 90-degree rotation back and 90-degree rotation, side and 90-degree rotation back. We can see what is happening. For example, in this chemokine recognition site 1, we see the flexible N-terminus of the receptor with its sulfated tyrosines interacting with the globular core of the chemokine with perfect charge complementarity. In the chemokine recognition site 2, we see why the presence of a glutamine at the very N-terminus of the receptor, of the chemokine variant is important. As it happens, glutamines spontaneously cyclize, N-terminal glutamines spontaneously cyclize to form pyroglutamates. And this rigid structure places in a very snug sub-pocket within the binding pocket of the receptor. And finally, the role of lysine 33, which was far away from the N-terminus in sequence, but not as far in the structure, is probably in properly positioning of the extracellular loop 2 of the receptor by this charge interaction. We also conducted a similar exploration for CXCR4 with several variants of its endogenous chemokine CXCL12. The first variant is wild-type CXCL12, and it's an agonist over the receptor. A single-point mutation that was previously known makes it into an antagonist, and a model of that interaction is presented on panel C. And finally, 2 more variants were developed in our lab. They are high affinity antagonists than even this one, and they were identified from a phage display with selection on live cells. So this work allowed us to draw some kind of structure-activity relationship. For example, we understand why a single amino acid extension on the N-terminus of the chemokine, in this case, it is beneficial for binding affinity because, as in case of CCR5, there is a snug pocket located here that residue can occupy and thus increase the affinity. So finally, this is a prediction. These are computational models, and people usually ask, okay, is there any validation of that? And in terms of retrospective analysis of data, these models are consistent and they explain lots of early and mutational studies in cysteine trapping that we and others performed. In case of CXCR4, it is also consistent with an X-ray structure of the chemokine with a small molecule inhibitor that was published recently. We also conducted some prospective validation studies, and that included selecting and testing additional residue pairs in cysteine trapping experiments. As this illustration shows, there is a pair of residues. These two spheres show the C alpha atoms of these residues. They are proximal and in favorable orientation to form a disulfide bond when they're mutated to cysteines. And indeed, when these two residues are mutated, we get the most stable and well-behaving complex. And in case of CXCR4, some functional experiments were performed with residues predicted to be important from the models. And they were confirmed to be important for signaling, as illustrated here. When D262 in the receptor is mutated, the complex loses its signaling capacity. So I will wrap up at this, and thank again my collaborators, and thank this audience for listening to me, and I will be happy to take any questions.
Okay, we have time for 1 or 2 short questions.
What happens to the structure when CCR5 is uncoupled? And there are some HIV variants that learn to recognize uncoupled CCR5. So is there any shift on the relevant positions? Did you ever look at this?
This is a good question, and from the structural perspective, understanding of activation and conformational changes associated with coupling of CCR5 to G proteins is not completely understood. We do have ideas of what is happening with GPCRs in general, what kind of conformational changes are expected, but there are no specific studies on conformational changes of CCR5. And it is important because some studies show that Moraviroc, for example, selects some conformations, but then others remain available for GP120 binding. And we're after that as well, but that's as much as I can say at this point. It's an open question.